Now let's talk about methylene blue reductase test or the MBRT test. Okay, this is a test which is commonly used in the case to check the quality of the milk. Okay, and actively growing microorganisms reduce the oxidation reduction potential of the milk medium because of the exhausted oxygen by the microorganisms. Normally, milk is contaminated by various microbes like Staphylococcus aureus, Streptococcus pyogenes, Pseudomonas aeruginosa, Enterobacter, Bacillus, and all. Okay, and as a result, what happens when they grow? What happens? The oxygen among the milk, the oxygen in the milk is which has been present in the milk will be found to be reduced because all these microbes will be utilizing all that oxygen which has been present in the milk. So what happens? The oxidation reduction potential of the milk will be found to be reduced because oxygen is already been used up by the microbes. And methylene blue reductase test is based upon this oxidation reduction potential of the milk. Okay, we will see how is it. Now this is a methylene blue reductase test which has been performed. Okay, this is a controlled milk which has been at three different time points. You have a different color. You take the milk, you keep it, add methylene blue, and after some time, what happens? The blue color gets degraded, and depending upon the time which it takes to degrade. Okay, depending upon that, the quality of the milk will be determined. So let's see how is it. The principle depends upon the fact that the color imparted to the milk by adding a dye such as methylene blue will disappear more or less quickly, which depends upon the quality of the milk to be examined. So methylene blue is a redox indicator and it loses color under the absence of oxygen, and thus it has been found to be thought to be reduced. Now, when the oxygen becomes depleted in the milk due to the production of reducing substances in the milk, due to the enhanced rate of bacterial growth, etc., what happens? This color gets methylene blue gets reduced at a very faster rate. Okay, the time taken for the methylene blue to get reduced will be found to be very less, and thus it indicates that the milk is not of good quality. But the dye reduction time, it refers towards the microbial load in the milk and the total metabolic reactions of the microorganisms. So if it takes a longer time for reduction, that is, a longer time has been taken for the methylene blue's color to go, it indicates that it is having a less microbial load and the quality of the milk is very good. Okay. Now, what is the procedure to it? You transfer 10 ml of milk into an appropriately labeled test tube and 1 ml of the redox indicator. What is the redox indicator here? Methylene blue. That is being added into the sample, and you tighten the test tube mouth with stoppers and gently invert the tubes about 4 to 5 times so that the proper mixing is happening. And then you keep the test tubes in a water bath at 37 degrees centigrade, and you must look the incubation time. You just can't leave it there and go. You have to at intervals of time you need to you check whether the color is being seen, and the time elapsed for the color to turn whitish appearance is noted. So, and you have to check the tube every 5 minutes. So, the result interpretation is in such a way, it will result in the reduction within 30 minutes if it is reduced. What is reduced? Methylene blue is reduced. If methylene blue is reduced within 30 minutes, it refers that the milk is of very poor quality. And if the reduction occurs between 30 minutes and 2 hours, it is of poor quality. And the reduction occurs between 2 to 6 hours, it means a fair quality. And if the milk becomes white only after 6 to 8 hours, it indicates that it is good quality. And even in 8 hours, if it is not reduced, that means what happens? It is an excellent quality. So, depending upon this, you will grade the quality of milk. That is, you take some milk and you add some methylene blue into it, and how long it takes to change from bluish color to whitish color, that has been noted. If in 30 minutes itself, what happens? See, you should keep in your mind, the change of the blue color to the white is mainly depending upon when this particular component, that is methylene blue, is getting reduced. Reduced means what? Removal of oxygen, that is oxygen is depleted. Only in oxygen depleted conditions reduction will happen. When that means if oxygen is being utilized, if it gets reduced at very fast, that means someone has used the oxygen in the milk. Okay, it's indirectly indicating that there are lots of microbes present in that. And if it takes lots of time for the methylene blue to become decolorized into white, that means to get reduced to white color, it indicates that there's lots of oxygen in the milk, and what is it? There is less amount of microbes in the milk, and that indicates that the milk is of good quality. I think this is clear to you now. So the disappearance of the color with limited time indicates that the absence of oxygen in the milk, and refrigerated milk contains more oxygen than the warm milk, and at the time of milking process it has more oxygen content than other cases. So the rate of reduction depends upon the nature of the organisms present in the milk. The rate of reduction by different organisms, like it has been arranged here in the order, like E. coli is greater than Streptococcus lactis, okay, and fecal streptococci is greater than micrococci. Micrococci is greater than thermoduric thermoduric organisms, and it is greater than the psychotropic organisms. What are thermoduric organisms? These are organisms which can withstand the pasteurization temperature. Okay. And the presence of light fastens the reduction rate, and hence the test tube under observation should be tightened properly. And use a uniform concentration of methylene blue in all the test samples, because if there is more methylene blue, it will result in more reduction time. So you make a standard procedure of how much of methylene blue has to be added. An increased incubation time reduces the reduction time, since the activity of some organisms increases with increased incubation temperatures. And periodically you should invert the test tubes at regular intervals during the incubation time, so that you get a better result. The results become more accurate, and it becomes more improved when you do such a thing. Otherwise what happens, at one particular area the decolorization will happen, but the other place it will not happen. Okay. And moreover, one more thing, the microbes might not be evenly distributed towards in the milk sample. So when you mix it completely, what happens, the microbe also become evenly distributed, and you will not get into wrong result interpretations. So that's all about the MBRT test, or the methylene blue reductase test. The next test at which we can use is a viable bacterial count to estimate the amount of bacteria which has been present in the case of milk. This we have already spoken with in the case of microbiological analysis of food. Like first you serially dilute the milk, and then you put it onto the plate. Either you pour plate it, or you put it into a spread plate method. Depending upon that, the sample of that you use also will be different. If you take pour plate, you will have about 1 ml of the sample is taken, and if you take a spread plate method, you take 0.1 ml of the sample. And for the dilution, we are using your 9 ml saline, or you can also use a Ringer solution, otherwise known as a Ringer solution, and you grade the milk depending upon the standard plate count method. Okay, less than 2 lakhs is considered to be as very good, and when it goes beyond that, the quality of the milk has been found to be less. The advantages, we have already mentioned this. The enumeration of only viable microbes have been present. In the case of other direct microscopic count and all that, you do not get to know whether it is a dead cell or whether it is a living cell, but in standard plate count method you can estimate only the living microbes. So that is an advantage here. And the cultural and the morphological differentiation can be done based upon the colony characteristics. Like if you have more than one microbe in that could also be identified by the standard plate count method, and it is suitable for the determination of the quality of pasteurized milk and even about high grade raw milk without low bacterial numbers. Okay. Normally in alcohol test and all that, what happens, like if it is, you're just, you get, what is it, when large number of microbes are present, you get a faster result. But in the case of here, what happens in standard plate count method, even extremely high pure milk like pasteurized milk or high grade raw milk and all, also the bacterial number will be very low, but even in that case also you can get a standard plate count method. Okay, and also for the line testing at various stages of processing of a pasteurization, that is at different stages you can take some milk and you can test it. Okay. The disadvantages is that it is time consuming, laborious and cumbersome, and it requires high quantity of reagents, chemicals and glasswares, and it is not a rapid method because at least 24 hours you have to wait for the result to come. That is, after serially diluting and plating it, you have to incubate a plate and only then you get the results. Okay. And the temperature of incubation is also a factor which is a disadvantage here, because different microbes have different ranges of temperature. So what happens, some microbes will grow at one particular temperature and the others will grow at other things. So that is a disadvantage, and sometimes you might many microbes may go unnoticed because of this variation in temperature. And the amount of the sample taken here is only a representative. I told you, you take either 1 ml or 0.1 ml. So sometimes if the milk is not properly homogenized or if it's not mixed properly, what happens, only a small quantity is taken and it is only representing a small quantity of the sample. And pathogens are not detected because certain microbes like Mycobacterium tuberculosis will not grow easily. You know, like Mycobacterium and all take 3 to 5 days for a single colony to come, and here we are waiting only for 24 hours for the standard plate count method. So Mycobacterium tuberculosis might not grow here. And another case what we have here is only in certain specialized media Mycobacterium tuberculosis will grow. We are not using a specialized media here, we are using a normal media, like let it be, if you are choosing bacteria, you choose a nutrient agar, and if you are choosing for yeast and malts, you take a yeast and malt agar or a PDA agar or something like that. So certain pathogens might go undetected when you use the standard plate count method as such. And specific information regarding the type of the microflora is not obtained in the case of standard plate count method. So with this we come to an end of this session. I hope you have understood all these tests of the milk analysis. We can have a better session with lots of queries. I expect that from you, hope so. You will come across with your queries and I can be able to answer it in our live sessions coming hereafter. Thank you.